This chapter we're going to talk about a process that is very similar to the process that we talked about in the last chapter, in chapter 10, that of DNA replication. This process that we're going to detail in this chapter, though, is called transcription. A number of times this semester we have mentioned the central dogma of genetics, and that is the flow of information from DNA to RNA and then from RNA to protein. We're going to start by talking a little, just sort of reminding you about that. We're then going to just take a general overview of gene expression. So gene expression is basically taking that genetic information and making it into something tangible. Most of the time that something tangible is proteins, amino acid chains, but not all of the time. Sometimes it's RNA. Sometimes RNA is the end product. The rest of the chapter will then be just sort of detailing this process and then pointing out a whole bunch of things that are different in this process when we look at prokaryotes and contrast that with eukaryotes. So once again, here's the central dogma. Read it, learn it, live it. So this is a figure 11.1 in the book. It's a nice little diagram showing you the basics of the central dogma of biology. It's broken up into two different parts. The first part, which is what we learned in chapter 9, is DNA replication. So if you see up there on the top, the perpetuation of genetic information from generation to generation, that again too, these are sort of these subjects that we keep touching back on. That was something that, if you remember, when we were talking about what did the genetic material need to do, it needed to be able to pass on information from one generation to the next. So at the top there, DNA replication, which is facilitated by DNA polymerase. We talked about the polymerases and things like that. And you go from one strand of DNA to two identical strands of DNA through that semi-conservative replication. On the diagonal is what we're going to be talking about. And so chapter 11 is the green part, that's transcription, where you go from DNA, or your information housed in DNA, to basically what amounts to a sort of a photocopy of that information. That photocopy is your mRNA. In the next chapter, we'll be talking about the translation of that copy of that genetic information from mRNA into an amino acid chain or a polypeptide. That's what we call translation. You see there in the red, reverse transcriptase. This is going or taking information from RNA and converting it into DNA. This is what happens with retroviruses, for instance, RNA tumor viruses. They will transcribe their RNA information into DNA information and then insert that into the cell's DNA or the genome. We'll start by talking about transcription in prokaryotes. This is a slide that shows transcription and translation in prokaryotes. It's fairly straightforward. The primary transcript is essentially equal to the mRNA molecule, so there's no sort of modification. And this will make more sense when we talk about it in eukaryotes, where you do actually have some processing and modification going on. The mRNA transcript then gets translated to your amino acid chain or your protein. So one sort of question, or something that we should probably address right now, is why do you do this? Why don't you just translate directly your information in DNA into protein? Why are you making a photocopy of it in essence? Why are you making a copy of it in essence? And there are a couple of different reasons for this. One, your information is valuable, right? If you just have one copy of it in your DNA and you're using that copy to produce your protein, it could get damaged. And so there's some safety in making a copy of it, and for instance in eukaryotes, shipping it out into the cytoplasm where it can then be translated. Also, you can produce multiple copies of it. You can also produce copies that then can be regulated through some other molecules and things like that. And so you can have better control of the final products or the rate at which you are producing these proteins and things like that. So this slide then shows you transcription and translation in eukaryotes, where, like I mentioned in the previous slide, you have a pre-mRNA molecule that then gets processed into an RNA or an mRNA molecule. The mRNA molecule then gets shipped out to the cytoplasm and gets translated. So as you can see, it's much more complicated. Both of these slides just show you the general process, and what we're going to do is we're going to spend the rest of the time sort of showing you these two general processes, but in much more detail. So before we get started, let's just throw out all of the RNA, the different types of RNA molecules that you'll need to know. The first three, messenger RNA, tRNA, ribosomal RNA, are sort of the classic ones that you probably all heard of before. The other two, the small nuclear RNA, are components of spliceosomes, which sort of splice the mRNA in eukaryotic cells, or the pre-mRNA in eukaryotic cells to create the mRNA. And then the microRNA, which are used for regulating gene expression. This is a really nice diagram. It's figure 11.2 in your book, and it goes through and just sort of shows you where each of those different mRNA molecules are kind of doing their thing, but then it also breaks it down for you in terms of what's happening within the nucleus and what's happening out in the cytoplasm. So if you start there on the upper left, you see your DNA molecule, you see transcription going on. This again is, obviously, since there's a nucleus, it's in a eukaryotic organism, in a eukaryotic cell. So let's just briefly sort of take you through the process of the production of each of these RNA molecules, and also maybe sort of take you through and show you the function of each one of them. And so let's start with the main one. So there you have your DNA molecule. As you can see in the upper left-hand corner, the molecule, and we'll go through the details of this, that you have a gene within your DNA molecule. The separate strands of the DNA separate through the workings of the RNA polymerase. You then transcribe your pre-DNA molecule. That pre-DNA molecule, as you can see up there, is sort of color-coded. It's, what is that, red and green. Those are your exons and introns, part of which will eventually be transcribed, part of which will eventually be excised out, or pretty soon will be excised out by that spliceosome. The spliceosome is an organelle that is found within the nucleus. The function of the spliceosome, just like the name suggests, is to splice apart the pre-mRNA molecule to form mRNA. The mRNA will then leave the nucleus through a nuclear pore, go out into the cytoplasm, be exposed to some of these other molecules that we're going to talk about here in a second, which are involved in translation. The main one, which is tRNA, which we won't get into too much detail about translation because that's for next chapter, but the tRNA basically brings the amino acid and brings the amino acids to the growing peptide chain, and that's where the translation is happening. That translation takes place in the ribosome, which is made up of rRNA molecules and proteins, and all of this is happening in the cytoplasm. So the other molecules, so rRNA there, or wait, let's do the S and RNA. Those are transcribed. So you go from DNA to RNA, those are transcribed, but then they become the end product. And so you have an RNA molecule that then associates with proteins, and kind of like with the ribosome, you form an organelle out of that association between the sRNA and the proteins. That becomes a spliceosome. The spliceosome then goes and cleaves off part of the pre-mRNA molecule that is not going to be translated. It excises it out and converts that pre-mRNA molecule into mRNA. Okay, the rRNA. So the rRNA gets transcribed from the DNA, then it finds its way outside of the nucleus through the nuclear pores and associates with the different proteins. They form these subunits of the ribosomes. The ribosomes then, like we mentioned before, go and perform their function, which is translating the mRNA molecule into protein. Then you have the mRNA molecule also going through the nuclear pore into the cytoplasm to be translated, and then the pre-mRNA, which goes through the nuclear pore out into the cytoplasm and then is modified or processed by a protein called the dicer, and it goes from pre-miRNA to miRNA, and that the function of that then is to repress translation or regulate gene expression. Central dogma again, and some of the generalities of transcription and translation. Okay, so let's start talking about the details of gene expression, going from DNA to RNA and then to protein. Okay, so here are some of the general features of RNA synthesis. Like I mentioned before, RNA synthesis is very similar. It's a similar process to DNA synthesis, but there are a few differences. And so up top there, we list some of the. You can see some of those differences. In RNA synthesis, the precursors are ribonucleoside triphosphates, and so that's a nucleotide. The difference between a nucleotide and a nucleoside is that a nucleotide is essentially a nucleoside with a phosphate group. And so here, obviously, with the synthesis of RNA, you're going to have, we'll just call them RNA nucleotides. Another difference is that only one strand of DNA is being used as a template. If you remember in DNA replication, both you had a lagging and leading strand. Here we just have one strand being used as a template, so the gene itself is just located on one of the strands. Another really important difference, and this gets to the difference between RNA polymerase II, which is the polymerase that is used to build mRNA or pre-mRNA molecules, and DNA polymerase III, is that the RNase does not need a primer, and so they can initiate without having that primer requirement. The RNA molecule that is either pre-mRNA or the mRNA molecule that results from transcription is going to be identical to the DNA complementary strand, the strand that's complementary to the template strand, only you'll have uridine instead of thymidine. So you have U's instead of T's in that strand. And then lastly, just like with DNA synthesis, you are going to proceed in this, you're going to have the same directionality. You're going to go or grow in a 5' to 3' direction. So here's just a diagram that kind of shows you what's going on. As you can see, sort of just to clarify some of the terms we use, we have a template strand and a non-template strand of DNA. Sometimes you'll see the sense strand or the antisense strand. I prefer just to use template strand and non-template strand. And so you have a template strand of DNA. That template strand gets transcribed into, in this case, we'll just pretend we're in a prokaryotic cell, gets transcribed into your mRNA molecule. Here they've labeled that the sense RNA strand. Again, we can just call it the template RNA strand. This slide shows you the molecular detail of the growing strand of RNA. It looks very similar to a figure that we had in the last chapter, only I want you to look at the sugar where you have the 2' carbon, right? So you still have the 5' and 3'. Those are kind of in the same places at the 3', and you also have that hydroxyl. But here, where it's different than DNA, is at that 2', so just to the right of the 3' carbon, you now have a hydroxyl rather than just a hydrogen. That's DNA is the deoxy. That means it's missing that oxygen. But again, you have guanine, cytosine with 3 hydrogen bonds, but now rather than the T, the thymidine, you have the uracil connecting or bonding through 2 hydrogen bonds with the adenine. And so it's very similar. It's growing in a similar direction or polarity to it. And so maybe a good idea would be to go back to chapter 10 and look at this same slide, but the one that we were showing you, the DNA synthesis, and just see how similar it is, and then also see what the slight difference is also. So even though RNA polymerase doesn't need a primer to begin transcribing that mRNA molecule, you don't want it to just start transcribing everywhere, right? You want some control of that. And the control of that happens or is signaled through a specific nucleotide sequences that we call promoters. That sequence, with the help of transcription factors, kind of signals where transcription should start. So here we see RNA polymerase attached to a DNA molecule. And RNA polymerase is nice because it does a whole bunch of things it performs a whole bunch of different functions. So RNA polymerase will come in there, it'll unzip the DNA, it'll transcribe the mRNA, and then within that same RNA polymerase, the DNA molecule will anneal, the 2 strands will anneal with each other. And so you basically have everything going on within that RNA polymerase enzyme. Key points to this section. RNA synthesis and DNA synthesis are similar in many respects, but also slightly different. So in prokaryotic cells, there's no separation between the DNA and the area where that DNA is being transcribed into messenger RNA molecules. It's all within the cytoplasm. There's no nucleus to separate those two happenings from each other. Just like with DNA replication, there's 3 stages of transcription. There's initiation, elongation, and termination. So initiation involves 3 steps. First, the binding of the RNA polymerase, the holoenzyme. That is a whole bunch of subunits plus the sigma factor. Second, the unwinding of the 2 DNA strands by the RNA polymerase. And then the third step is the formation of bonds between the first few ribonucleotides in the beginning or the nascent RNA chain. After 8 or 9 of those nucleotides are bound, the holoenzyme releases the sigma factor. And you then enter into the elongation stage. Where the initiation begins depends on several factors within that promoter region. This is a diagram. This slide shows a diagram of just some generalized characteristics. There is variation in this within prokaryotic organisms, especially between true bacteria and archaebacteria. But this is just a good generalized depiction of it. So if we look at the promoter region, we see that there are two chunks of it that are fairly conserved amongst sort of lots and lots of promoter regions. One of them is located at -10, we call it the -10 box, another one is -35 box. If you look just above that, the DNA molecule, you see a -1, -10, -35, and a +1. Basically, that is telling you what is upstream and what is downstream of where the molecule begins to be transcribed. So -1 means it's one base pair upstream, +1 means it's one base pair downstream, -10 means it's 10 base pairs upstream, -35, 35 base pairs upstream. Also, there is sort of the distance between these two chunks of the promoter that are fairly conserved. There is some variation in this, but it's pretty well conserved between 16 to 18 base pairs. I actually read even up into 20 base pairs, but just that not necessarily the base pairs that are located within that, but just that distance is a fairly conserved distance. And so the -10 box and the -35 box, -10 box consists of nucleotides TA, TA, AT, and then in the -35, TTGA, CA. Okay, as you can see here, especially in the -10 box, made up of lots of T's and A's, where you're going to only have two hydrogen bonds between the nucleotides. That makes it easy to pull apart and to initiate the reading of the template. Once the RNA polymerase, like I mentioned before, releases that sigma factor, we then jump into the elongation stage. Here the RNA polymerase unwinds and rewinds the DNA molecule, and in between those two things, it adds RNA nucleotides to the growing chain. In prokaryotic cells, this can be one gene, one complete gene, or it can be several genes. In E. coli, the average length of the transcription bubble is about 18 nucleotide pairs, and it adds nucleotides at a rate of about 40 nucleotides per second, so it's fairly fast. Once the RNA polymerase transcribes the mRNA molecule, we then enter into the termination stage. Sorry, I spoke too soon. We are still elongating. Now we're ready to terminate. In prokaryotic cells, there are two types of terminators. There are Rho-dependent terminators and Rho-independent terminators. The difference is that the Rho-dependent terminators require a protein factor that we call Rho. We're not going to talk too much about the Rho-dependent terminators because they're very similar to the Rho-independent terminators. They just have this protein factor that's involved. So let's just move on and talk a little bit about the details of the Rho-independent termination. So termination happens when the RNA polymerase encounters a termination signal. Here we're looking at Rho-independent termination, and I want you to look at the sequence of DNA that is highlighted in blue and then eventually yellow, but specifically in blue. What do you notice about it? After looking at it for a little while, you should see that they're inverted repeat sequences. Functionally, what this means is that once the mRNA transcript is made, you can see there on the bottom, the sequences that are produced are going to bind with each other and form this structure that we call a hairpin. This hairpin structure forms immediately after it is produced, and what it does is it causes a pause in the chain extension. During this pause, now let's look at the sequence in yellow. You see up top it's AAA, and then down at the bottom it is UUU. Since the AU pair bonding is weak, it requires less energy to separate the bases. And so essentially what results from this is the RNA polymerase freeing itself from the DNA strand and releasing the newly manufactured mRNA transcript. Lastly, we should note that since this is in prokaryotic cells, we can have transcription happening along the DNA molecule. Even before the transcription is terminated, you can have ribosomes coming in, latching on, and you see that on the right of this electron micrograph picture. You can see the ribosomes latching on to the mRNA molecule and translating. So you get transcription and translation not separated in space like you see, or like we will see in eukaryotic cells. So here's a really cool link to a video on YouTube that kind of shows you this whole process in a sort of a consistent scenario. So you can kind of see what's happening in real time. Key points about general transcription and some aspects of prokaryotic transcription. Transcription in eukaryotic cells is similar as that in prokaryotic cells, but it is a little bit more complicated. For instance, there are 5 different RNA polymerases present in eukaryotes, and the transcripts undergo 3 major modifications prior to their transport to the cytoplasm for translation. One of which is splicing out introns, or non-coding chunks of DNA or RNA. Here are 3 major differences. Here are the 3 major modifications of the pre-mRNA molecule. The first one is the addition of a 7-methyl-guanosine cap to the 5' end. Essentially what this cap does is it allows the protein factors involved in the initiation of translation to recognize the mRNA molecule, and it also helps protect the growing RNA strand from degradation by nucleases. The second major modification is the addition of a poly-A tail to the 3' end of the transcript. The poly-A tail enhances the mRNA's stability, and it plays a role in the transport of the mRNA or pre-mRNA molecule from the nucleus into the cytoplasm. And the last modification is the splicing out of the intron sequence. And we'll talk more about introns here in a second. Here's a diagram that just sort of graphically shows those 3 modifications. Just like in prokaryotic cells, eukaryotic cells go through an initiation and elongation. and a termination. The specific polymerase that is involved in the formation of the mRNA transcript or the pre-mRNA transcript is RNA polymerase II. But eukaryotic RNA polymerases cannot initiate the transcription by themselves. They actually require the assistance of protein transcription factors to start the synthesis of the RNA chain. Here you see several kinds of transcription factors binding to that promoter region at the different locations upstream from the transcription start point. Next, transcription factor 2F binds and has an unwinding activity to it, so it begins to unwind the DNA molecule. Then transcription factor 2E binds to sort of complete that initiation complex, with RNA polymerase II coming in and joining the complex. Then elongation proceeds in pretty much the same way that it does in prokaryotic cells. Early in the elongation process, the 5' end of the transcript is modified by the addition of that 7-methyl guanosine cap. This slide shows the molecular composition of that cap. So in eukaryotes, the transcription actually goes beyond or proceeds beyond the site that will eventually become the 3' end of the transcript, the 3', what we call terminus. And in fact, that terminus or 3' end is produced by cutting the transcript through, as you can see here on the slide, through something that we call the endonucleolytic cleavage. So it'll actually proceed beyond that and then cut it. After this cleavage, the enzyme poly-A polymerase adds the poly-A tail. Normally, the genetic information contained in the mRNA molecule is not altered, but we recently discovered that in fact some RNA editing does take place. So here I just sort of mention this exception to the rule. Okay, so most eukaryotic genes contain both coding and non-coding sequences. The coding sequences are called exons. The non-coding sequences are called introns, and the introns have to be removed from the pre-mRNA prior to them getting shipped out to the cytoplasm. We'll talk more about the exons when we talk about translation. So now let's just sort of focus on some of the details of the introns. The word intron comes from the phrase intervening sequences. Again, they're located between the exons or the coding sequences, and they actually can be quite variable in size, some of them being very, very large. One interesting thing from sort of a molecular biology perspective, or an evolutionary biology perspective even, is that within those genes you have introns and exons, and they, even though they're sequenced within the same gene, they are going to be exposed to very different evolutionary pressure. The exons will have, a very, very tight selection against them, right? I mean, that you're not going to see a lot of rapid changes in the exons, because a mutation is most likely going to be deleterious. But within the introns, they are sort of freed up to evolve and accumulate mutations, and so you can use that when you're trying to figure out the relationships between organisms. You know, you can use that fact. But again, prior to the pre-mRNA becoming mRNA, you need to remove those introns before it heads out to the cytoplasm. So the obvious question sort of becomes, why have these introns? Why have these regions of the gene that are not going to be translated? And there's been a lot of discussion about this, a lot of research, and there seems to be a couple or a few ideas being debated out there. First, because of the variable size of introns, people thought, oh, okay, maybe that has something to do with regulating the gene expression, because if you have to take time to transcribe an intron that's really long, it's going to take longer for the whole process to come to fruition and have that transcript modified and then sent out to the cytoplasm to be translated. Whereas if you have short introns, you can do it quicker. Also, recently there has been some research that shows that some introns contain sequences that can actually regulate the gene expression either in a positive or a negative fashion. Another idea is that the increased rate at which the coding sequences in different exons of a gene can reassort by recombination. So you can, in that sense, it opens up the gene for creating recombinations of the actual structure of the gene, and that allows it to evolve in a quicker fashion. The last idea is that if you sort of have different chunks of genes, possibly you can construct them in different ways to give rise to genes or combinations of chunks of genes with new functions. So now the question becomes, functionally, how are these non-coding introns removed from the pre-RNA transcript? And what we've found is that there are a few different mechanisms. As you can imagine, splicing out introns has to be very, very precise, because if you cut out an intron, so let's say just for example intron A up there, and you miss it by one nucleotide, you're going to mess up how the resulting mRNA is read. And in a sense, it's kind of like mutations. Back in 1110, you probably talked about frame shift mutations, where you shift one base pair and it messes up everything downstream from that. So the same sort of principle is true with splicing out introns. If you're not really, really precise about it, you could end up with a product that's really, really messed up. It doesn't seem like there are a lot of conserved sequences present in the splicing process here. We just kind of mentioned too. So with this, don't worry too much about all of the exact detail of the splicing process, but what I do want you to notice is that you have these SNRNPs here that are binding to the splice site. The SNRNPs are small nuclear ribonucleic proteins, so it's a combination of proteins with the snRNA that we mentioned earlier in the chapter. And so you have them binding to the site, going through this process of assembling into a complete spliceosome, then bending the transcript and eventually cleaving the sites and joining them together, where down at the end you now have your spliced RNA transcript. So again, not too much detail on that, but just kind of understand what's going on. Okay, so that is it for transcription. There are a couple of other kind of splicing processes that I'm not going to really talk about. You can read about a little bit. There's a couple of paragraphs in the book about them. One is the tRNA precursor splicing, and the other is the autocatalytic splicing. But we're not going to concentrate too much on those. Okay, so here are the key points for this section. And lastly, I just want to stress again how precise it has to be. It has to be down to a level of accuracy of a single nucleotide. You can't shift one way or another in that splicing, otherwise you are going to pretty much have an mRNA transcript that isn't going to give you the product that you need. Okay.